And joining us now on the debate for the full hour tonight, in Seattle, Washington, Leroy Hood, a pioneer in the area of genomics and biotechnology. He's president and co-founder of the Institute for Systems Biology. In Houston, Texas, Amy McGuire, associate director for research in the Center for Medical Ethics and Health Policy at the Baylor College of Medicine. And with us here in studio, Dr. Catherine Siminovich. That's harder to say than I thought, isn't it? Siminovich, immunologist who directs the Adult Clinical Genetics Service at Mount Sinai Hospital. Dr. Lillian Chu, a medical oncologist and co-director of the Drug Development Program at Princess Margaret Hospital in Toronto. And Alison Symington, Vice President of Outreach at the Ontario Genomics Institute. That's a not-for-profit corporation focused on bringing genomic research to the marketplace. Okay, welcome everybody here in the studio. And to our guests in the United States, this is a most fascinating topic, but my suspicion is a lot of our viewers need some of the basics. So we need a basic primer off the top, and Alison, I'm going to go to you first on that. We talk about personalized medicine. What's personalized medicine?
Well, really, personalized medicine is just using your genetic material and the environment to determine or manage your healthcare. So the idea is that we'll be able to screen people's DNA and be able to give that information to patients and their physicians to manage healthcare outcomes, better drugs, potentially predict what risk factors they may have, so that overall we can use it in both a preventative and a predictive way around healthcare.
Okay, so Leroy, looking at its full incarnation down the road, what will the practice of personalized medicine look like as you imagine it compared to, say, how healthcare is practiced today?
I would say there are two aspects to personalized medicine, and parenthetically we'll get into this later. I call it P4 medicine because there are many dimensions that extend beyond personalized medicine. But I would say number one, it's going to be all about quantifying wellness. That is, as we move forward more and more, we're going to focus on wellness rather than disease. And number two, it's going to be using enormous amounts of information. I would argue in the future, every patient will have a virtual cloud of billions of data points to be able to demystify whatever diseases they might have and fashion appropriate treatments for each.
Catherine Siminovich, how many years down the road does that vision start to really take shape?
I think we're approaching that vision in an incremental way right now, but it's going to be many, many years before we actually reach what Dr. Hood was just talking about. The technology has moved much, much quicker than our capacity to understand it. So we need to now use this incredible technology and information that's starting to pour out and learn how to apply it to healthcare.
When you say many, many years, we're talking decades, really?
I think we're looking at decades,
Decades.
Yes.
Because it's already been around for one decade, and I think everybody, well, certainly lots of people were hoping, thought we'd be there by now.
We are there. The technology has moved incredibly fast. So as was just said, we can get all the little genetic pinpoints and information about each individual, but understanding how those connect to specific diseases, how those connect with the environmental factors, takes a lot of research. A lot more work is needed.
Amy, I'm going to put some background information up right now, and then I'm going to ask you to comment on some of that with another question on the way out. Michael, let's put this background info up if we can. The human genome was meant to create a blueprint of the human body by determining the order of 3 billion units of DNA that make up our genome and to identify all of the genes, determined to be about 22,000 in number, incidentally. The project began, we're going back to 1990 now, and the projected cost at the time was $3 billion. The first draft of the human genome announced in the year 2000, and it was completed in April of 2003, and it has led to the discovery of more than 1,800 disease genes so far. Now, Amy, some notable people were the first to have their genomes fully sequenced. We know Craig Ventnor, of course, the scientist, did it. James Watson, Steven Pinker have done it. Since then, any idea how many people, let's just say in your country, have had their genomes mapped out?
I don't know exactly how many people have had it done because what you term whole genome sequencing is defined differently by different people, but certainly the technology has gotten to the point and the cost has dropped to the point where whole genome sequencing is relatively economical, and it's being used more and more in the research context. So I would say more and more people are getting their whole genome sequenced, at least in a research context.
Let me follow up on the cost angle, because presumably, you know, when this first started, we were talking at tens of thousands of dollars to get it done. What can you get it done for today?
Well, when we were first talking about it, we were talking about millions of dollars to get it done. And I would say today you can get it done for, depending on how you define the whole genome and at what coverage you're doing it, but you can get it done for less than probably $10,000 to get a good read.
And why would somebody want it done?
Well, I think there's different people who are motivated by different reasons. I think some people are just interested and curious to see sort of what's in their genome and what information we can glean from it. Other people are just really anxious to be part of the science and enthusiastic about moving the science forward, and so they don't necessarily want to know everything that's discovered in their genome, but they're just excited to be part of the research and to donate and contribute their DNA to advance science.
I see. I wonder if I could ask our director, Michael Smith, to take a wide shot here so that we can see all our guests here in Toronto. There we go. Thank you, Michael. And our guests out of town as well. Hands up. Who's done it? Who's had their... I don't see any hands going up. None of the 5 of you has had it done? Lee, how about you?
Well, I didn't do it for a number of political reasons, but my entire family has agreed recently that we all should get it done. So we'll probably be having it done within the next year or so. I would say that there are enormous advantages for sequencing your genomes within the context of your family. You can correct error rate, you can identify rare variants, all sorts of things much more easily. So having family genome sequencing is the way it's really going to be done in the future.
Let me just follow up on the first thing you said, though. You said there were political reasons why you personally didn't get it done. What did that mean?
Well, it meant that A, I developed the automated sequencer that made the genome project possible, and B, I played a role in getting the genome project started in the early days, and then had one of the NIH centers that sequenced the genome. And there were a number of the companies that came to me and said they'd do it for free, and I decided on principle I didn't want to have it done that way. And I'm really glad, because we'll have family genome sequencing, another form of togetherness.
Gotcha. Okay. Lily and Sue, how come you haven't got it done? You folks know this stuff, you know, as well as anybody around, and you haven't done it. How come?
Maybe I don't want to know what will happen to me. I think it's something in the future. If it happens, it happens. I think if I do develop cancer, as I'm an oncologist, I would want to know my cancer's genome. That may help me direct therapy towards my own illness. At this point, I'm fairly healthy. I'm happy with the way it is.
Gotcha. Okay. You spoke about the future, and to that end, Hollywood has got its claws into this story in a big way. And I don't know how many of you have seen the movie Gattaca, but it came out, I think, several years ago. And we're going to play just a little clip from that movie because it'll help set up a bit of a discussion to come. So, Michael, if you would, from Gattaca, roll tape.
I'll never understand what possessed my mother to put her faith in God's hands rather than those of her local geneticist. 10 fingers, 10 toes, that's all that used to matter. Not now. Now, only seconds old, the exact time and cause of my death was already known.
Neurological condition, 60% probability. Manic depression, 42% probability. Attention deficit disorder, 89% probability. Heart disorder, 99% probability. Early fatal potential. Life expectancy, 30.2 years.
30 years.
Catherine, that's Hollywood,
Right.
but how accurate is that?
Well, we're really not there yet. So what we've learned a lot, both by genome sequencing and even other genetic strategies now, is how to find genes for what we call the single gene diseases, like cystic fibrosis, where one genetic error, if you find that error, you know the person will have the disease. What we're trying to do with all this genomic information we're getting now is find the genetic information for common diseases, like the diseases you just heard about. Common cardiac diseases, heart attacks, etc. We have insufficient, really insufficient information at the current time to make those kind of predictions.
Do you think, though, that we are, I mean, how far away are we from a time where you could look at a drop of blood and say, oh, we know what's going to happen to this person over the course of their life and how long they're going to live?
I reiterate what I said before. I think we're decades away from that.
Decades away from that, okay. Amy, as an ethicist...
You know, can I break in here?
Sure, go ahead,
I
Lee.
would argue that we'll never be there. And that is because organisms actually have two kinds of information. One is your digital genome, that you can know more or less completely, but the other are the environmental signals that come from your the outside of the genome. And I would argue you'll never know what your environment is going to be, and the environment plays a critical role in the determination of who you are as well as how long you're going to live. So I would argue, there may be random kind of events that will occur as well. So I don't think we're ever going to be able to precisely know when a person will die.
Alison, what do you say on that?
I would totally agree with that. I think, you know, environment is dynamic too, not just a genome, which is also a dynamic thing. So the combination there, and I mean, I think that's maybe why a lot of people haven't gone and got their genome sequenced, because they get the information back and we're not sure exactly what it means. Gattaca's interesting because it talks about the probability. Well, what changes that probability? Well, that's the environment. And we don't have good information yet on what those influences are. So, you know, we don't know all the things that are going on there. So I think Lee's right, probably we'll never know, because that's always going to change. And perhaps personalized medicine would change that as well.
Lillian, do you find in your experience with the people that you deal with, we want to know, and we're hoping you're going to tell us?
Yeah, I mean, I think we're closer than we were 2 decades ago, 1 decade ago. We're making progress. I think I'm an optimist because I'm a scientist, I'm a clinician, and I totally agree with the panel that we're not going to be able to give the information that everybody wants 100% now, but we've made some baby steps, and that's important. That will take us to the next baby step. And I think we are making that kind of improvement day by day.
Amy, I'd like to ask you the ethical question here, which is whether or not you're worried about the proliferation of gene sequencing going on in your country, and to a lesser extent ours nowadays. What do you think?
Well, I think it depends on what people do with the information, and I absolutely agree that one of the problems with the movie Gattaca is that it vastly overemphasizes the role that genetics plays in sort of determining your future health state. And so if people misinterpret the information that they're getting and think that it's deterministic in a certain way, so if I have this particular variant, I'm going to get this common or complex disorder, like I'm going to die of a heart attack or something, which we know there's a lot of risk factors that go into determining, you know, whether you're going to be susceptible to heart disease or not. And so if people misinterpret that information and act on those misinterpretations, then I think it becomes a problem. But as long as people understand sort of the role of genetics and the fact that it's just one piece of the puzzle in determining sort of what your future health is going to look like, then I think it's useful information to have, and it can help inform decisions.
Amy, let me stay with you for this. You know, there are companies out there that do direct advertising and direct pitches to consumers, basically saying, you know, send us a saliva sample and we'll give you a good chunk of what, you know, of what your genomic readout is. What do you think of that practice?
So those companies are fairly controversial right now for a variety of reasons. And one of the complicated things with those companies is that they offer a whole range of genetic information. So some of the information they offer is really interesting information about your ancestry and tracing back your ancestry, and a lot of people are very interested in getting that information. They offer some information that they call sort of recreational information, so they may tell you based on sort of the latest science what your genetic likelihood of getting genes that say predispose you to being a fast runner or something might be. So those don't have particular health implications, but they may be fun to get. You probably know how fast you run if you've ever gone out and tried to run before, so it's not particularly informative, but it might be fun to get. So I think that the companies, though, start to get a little bit more controversial when they start offering information about disease risk. And again, there's great variety in the type of information that they're offering. They're offering some information about these common and complex disorders that we know are not caused by one single genetic change. And so it just may slightly increase your risk or decrease your risk of getting a certain disease like cancer or heart disease or diabetes. But then they're also starting to offer genetic testing for more single gene disorders and even complex disorders that might be more predictive of particular diseases. So some of the companies are offering results of people's genetic predispositions to getting breast cancer, and they're looking at the BRCA genes, which happen to be very predictive of your risk of getting breast cancer. So it's very difficult to talk about sort of the ethics of what the companies are offering because there's such a wide range in the information that you could be getting back. And the most important thing is that consumers are aware of the range of information and make sure that they want all that information before they sign up to get the results.
Sure. Let me get Allison to follow up on that. Can you give us some sense about the industry that is building around personal genomics today?
So there's quite a large industry, of course, mostly around the diagnostic tests. So being able to determine whether somebody's high risk for a certain thing or whether in fact a drug will work. So I think we need to also just go back, when we talk about personalized medicine, we're not talking about going in and fixing a gene. We're making a prediction based on a genetic pattern, whether a particular drug would work or in some cases, even as importantly, whether it won't work for you. So, for example, the one they just mentioned, which is the breast cancer gene, there are some drugs that work when you've got a certain marker, what we call biomarkers, so a marker that suggests that you have a gene, specific gene. So there's certain drugs that work better. So there's tests out there for a certain marker for certain types of breast cancer.
Just so I'm clear, is this before you've actually been diagnosed with breast cancer?
No, in that case, well, certainly in Ontario, the case, the way that's done here is when you're already high risk for breast cancer. We also have to look at it for those diagnostic tests in the realm of our healthcare system. So it's a whole other question there. But there are there are diagnostic tests for things like macular degeneration. There are certainly a whole slew of genetic risk tests for these single gene diseases that run through families, and certainly the huge genetic group at Sick Kids, for example, looking at those kinds of things. But there are also tests for things like coumadin, for checking what your blood level should be for coumadin, and there are genetic risk factors about how they should do that. And this saves people ended up going to hospital with adverse events they never needed to have, or taking a drug that never would have happened. So, or never, they should have never taken because it's never going to work for them. So I think when we talk about personalized medicine, we have to be a little careful not to talk about we're going to fix genes or do those things. We're actually talking about putting people in subpopulations, saying, this group of drugs will work better for you than this group of drugs. Not that everybody will have one, each will have our own drug designed for each one of us. That will never happen because the industry would never make money.
Designing an individual drug for each
and
person.
ultimately, diagnostic kits make money, made by companies to make money, so that there is that whole industrial part about it too.
Let me follow up on something, Lee, you said right off the top of the program. You talked that acronym, P4. In the way, I guess, referring in the way to the way doctors are going to change the way they practice medicine. Why don't you, spell out the P4 argument for us, if you would?
So the P4s are the following. Predictive, I would argue in 10 years, many of us will have our genomes done, and that will provide incrementally larger information than we can get from them now. I would argue that we'll have a little handheld device that can prick your finger and measure 2,500 blood proteins and be able to assess wellness as opposed to disease in your 50 major organs. The personalized side is really fascinating because we're all genetically different. We have to be treated, as we've implied, as unique individuals and not as populations. That means each patient is their own control for any transition from wellness into disease. But more than that, it means that with billions of data points for each individual, we'll be able in time to sculpt your wellness and disease with exquisite precision. And of course, what is really exciting is with hundreds of millions of genomes and their environmental data, we'll be able to mine for the predictive medicine of the future. Prevention is about completely new strategies for designing drugs using network biology and drugs that can make disease-perturbed networks behave in a normal fashion. But what prevention is really about is wellness, and we'll get metrics for wellness. We'll all be thoroughly engaged by these wellness and thoroughly engaged in fashioning our own future health history. And the fourth P is participatory, and that really relates to the social challenges of P4 medicine. How do we train the patients, the physicians, the members of the medical community? How do we create an IT for healthcare that can deal with billions of data points and reduce that enormous dimensionality to simple hypotheses about health and disease? So those are the 4 Ps.
Gotcha. Well, predictive, personalized, prevention, and participatory. And Catherine, I wonder how many of those 4 Ps,
Are we there yet?
I don't want you to think you're always the person I come to with the "Are we there yet?" question, but are you doing this?
Yes, for example, for the single gene diseases as an example already, and I used the example of cystic fibrosis before, we know the gene, we know the kind of lesions we're looking for. It's possible to take someone and look at their DNA and predict whether or not they're going to have that disease. But even the way you express that disease is also influenced actually by factors in the environment, so we have to be careful in making predictions with that information. For the more common, complex diseases, for most of those diseases, almost all of them, we just don't have enough information to be making predictions yet. We have little pieces, and that's why, in a way, what these companies are offering is a little difficult to handle, because they make predictions based on incomplete information. So the consumer that's looking at has to know the information is very incomplete. Don't run away and think that this is telling you you're going to get Crohn's disease or rheumatoid arthritis. We absolutely cannot predict that.
Potential for overreaction is really quite significant, isn't it?
That's what I worry about in that. And in our clinic, we already have people who have signed up and got information from some of these companies in the States, and they come with these papers and show us the information and say, well, what does that mean? And some of them are very nervous because they see words like, say, Crohn's disease or some kind of cancer, and they get very nervous with the information. And it's kind of unfair, because really, if they look at it, if you look at the information carefully, what the company is saying is a tiny, tiny probability number that's truly, in the scope of everything, meaningless.
I know Alison touched on this a moment ago, Lillian, but let me get you to follow up as well. How much are we relying on genomics now when it comes to treating cancer?
Cancer is a very complex disease, Steve. So at this point, I think we're still at the research stage. For example, at Princess Margaret Hospital and many big cancer centers in the world, there's active research effort trying to understand how we can target the cancer genome and then match specific populations, like Alison said, to these drugs that are either in development or being developed, so we can give them treatments that we hope can help them for a period of time and refrain from using these drugs in patients who won't benefit. But the research is still ongoing, and you have to prove something like this through clinical trials. You can't just subscribe the drug or prescribe the drug immediately when you think there is a mutation. It has to be valid. It has to be proven.
And Alison, we hear this expression gene therapy used all the time. What does that mean?
So gene therapy is a little different than personalized medicine. Gene therapy is when you can go in and correct a genetic I don't want to say problem, but it might be a difference. It could be an overexpression, it can be an underexpression. Quite often, many of our current diseases are a problem like that, like cystic fibrosis, well, cancer. In fact, most of us would argue many of our diseases are a combination of environment and genetic, so they could be theoretically changed by gene therapy. So this is the scare that everybody has around genomics, that we'll be able to take everybody's sequence and then we'll be able to manipulate the genes to make things, and gene therapy could help people. And so the typical one, when I'm telling people about gene therapy, and the one you'll hear out there the most, is kind of the boy in the bubble disease. So the boy in the bubble, there was a movie a long time ago, John Travolta, I remember that, but there's also a Simpsons episode. But anyway, so the boy in the bubble disease, where he has no immune system, must live in the bubble. And so that is a genetic defect, and there has been gene therapy to treat that, and it has worked in some cases. But we're very early on in gene therapy, and so it's also not worked in some cases where we've perturbated the genomic system and had bad things happen, things we didn't expect. But personalized medicine really isn't about gene therapy. So gene therapy is kind of another area that's very important. Lots of trials going on across the world in gene therapy. But it's not what we would consider everybody to go in, instead of saying you have this disease, we're going to genetically fix you. We're still looking at drug therapy when we're talking about personalized medicine.
It may not have to be that significant. Amy, let me talk to you about the ethics of, say, changing, you're going to have a child, changing the child's eyes from brown to blue or something like that. Is that something that gene therapy speaks to, or having the genomic code can help with?
So gene therapy typically deals with making a change once a child is born. And it's much more difficult to do that. But what is happening and what people are doing is they're doing pre-genetic diagnosis. So you can, if you do in vitro fertilization, where you fertilize an egg outside of the woman's body, and then you create embryos, which is frequently done for people who have fertility issues, it's possible to do genetic testing on the embryos, and you can look for a variety of different things, from disease genes to traits like eye color eventually, and you can look and sort of pick the embryos that you want to implant then in the woman. And so that's fairly controversial, because there is concern that people are going to make decisions about the type of children that they want to have based on factors that some people find morally objectionable to base childbearing decisions on, like eye color and hair color. But we're using it now for really deleterious genetic conditions to try to not implant embryos that have those genetic predispositions. And they're also starting to use it for some sex selection. So you can, in some cases, and the laws vary from state to state whether you can do this, but in some places you can choose the sex of your child through pre-implantation genetic diagnosis.
Alison, a follow-up?
Yeah, I'd just say in Canada, sex selection is not allowed unless it's in the case of a sex-linked disease that is very specific, haemophilia being an example. But it's not allowed in Canada. It's against the Assisted Human Reproductive Act.
Gotcha. Let's move into some discussion about the pharmacogenetic advances that have been made. And Lillian, I want you to start by talking to us about Herceptin. What is that?
Herceptin is a anti-cancer drug that specifically targets a group of cancer that shows a molecule called HER2 on their cancer cell surface. It's a drug that has been approved for the use in breast cancer. It's one of the poster trial drugs for personalized medicine because it specifically helps this group of breast cancer patients who express this molecule on their cancer cells. And in early advanced cancer patients, or in early stage cancer patients with breast cancer, for example, Herceptin has shown to decrease the risk of recurrence by about 50%. So I think that's why it's been used as being a drug that represents personalized medicine, or the concept of personalized medicine.
So it's better than radiation, better than chemo?
In addition to.
In addition to.
To these conventional treatments.
You wouldn't do it instead of?
Not instead of, in combination with.
Huh. How expensive is it?
It's expensive.
Okay, meaning what?
I actually don't know the exact dollars, but it is, obviously an expensive anti-cancer therapy in the hundreds and thousands.
Hundreds of thousands of dollars.
But that's why
hundreds to thousands.
Hundreds to thousands.
But that's why it's really important, and that this is a great. Pharmacogenetics is probably one of the most quick things that is going to come out in personalized medicine, and one of the things where you can really see immediate benefit, because this drug is so expensive. But it only works in a subclass of patients, and you want to know which. You don't want to give this to all breast cancer patients. It'll be useless in one class of them. But by genetically typing the tumor, you can figure out which. You can predict ahead of time in this subclass of patients, this drug is going to work with certain high percent efficiency, and that's true for. We're finding more and more drugs that are like that, and we've all known for years that we take patients, we treat. 100 patients with our first-line therapy, and only 60% of the patients respond. And wouldn't it be terrific if ahead of time we'd already known that the other 40% is not going to respond, and immediately give them something else? And I think that's one of the first real benefits we're going to get in the clinic from all our genomic information.
So Lee, tell us, does the math on this add up then if we're only talking about subgroups of patients who might be eligible to be helped by this?
Well, I'd make one further point about pharmacogenomics. Just as you can identify subgroups or stratified groups of patients where drugs work well, you can also identify groups where drugs don't work well. For example, there's a blood thinner called warfarin, and there are certain genetic defects that either confer on patients the ability to get the blood way too thin or the blood way too thick. So it turns out that there are perhaps of the order of 50 mutations or so that actually lead to adverse responses to drugs. So that's something certainly every patient that was going to take the relevant drugs would really want to know. Now, your second question is, I think, one of the foundational principles of personalized medicine. I think one of the first things that we have to do when we're dealing with ordinary diseases or complex genetic diseases like diabetes, like Alzheimer's, is we have to be able to stratify the patients so that we can identify the different types of each of those diseases. We have no idea how many diseases there will be, but that stratification is really critical because, one, without it, we can never understand the different mechanisms that lead to a similar set of diseases we now call Alzheimer's. But even more important, number two, there are probably 100+ drugs out there, and none of them work very well because you attempt to use the drugs on all the different patients. So we have to be able to find to stratify the patients and then do a proper impedance match back against the drugs. And there are very powerful tools that are emerging now for the stratification of disease at the genomic level, at the blood proteomic level, and even in using induced pluripotential cells from patients to help stratify their diseases.
Amy, let me do this follow-up with you, though. Do you, in your experience, your knowledge of Big Pharma, are they prepared to deal, the math of it, the economics of it? Are they prepared to deal just with subgroups as opposed to the whole mass market?
Well, it actually can be quite economical because if you think about it, pharmaceutical companies spend millions, sometimes billions of dollars trying to develop a drug. And if you're studying it at population level and it's only effective in a very small subpopulation, the drug may never make it to market because it doesn't have the effectiveness data that's necessary to make it to market. But if you can show that it's effective in a smaller group, then it may actually make it to market and become a profitable drug for that subpopulation. So the idea is that it will be an economical venture for pharmaceutical companies because it'll help them save drugs that otherwise wouldn't have made it to market.
Lillian, have you learned anything because of Herceptin?
Yes, absolutely. I mean, we know that cancer is a complex disease, and as you heard from Dr. Hood, from Lee, that this is going to be the future's formula. This is how we understand more about cancer. There are different subgroups of cancers, and we need to target specific subgroups with specific drugs. We also know that even if we can target these drugs with personalized medicine, they will eventually develop resistance. I think no matter what we do, there's not going to be a drug that would cure a cancer indefinitely. We will start needing to learn how to overcome these resistances to these targeted drugs. And I think we are learning every day based on what the genomicists and the proteomicists tell us, the new knowledge that we can advance our field. And
I know some people watching probably, I'm trying to remember, I think in Ontario, if you're a senior, your drugs are covered. If you're on welfare, your drugs are covered. If you're working, a lot of people have a drug plan at work. I think 85, 90% of Ontarians have some kind of, good comprehensive drug coverage. Is Herceptin covered, do you know?
For certain groups of patients, yes.
Only
but
for certain groups.
For certain diseases where this drug has seen some to be beneficial, it's not always covered. And that obviously is a barrier.
Gotcha. Okay, let's do a little reality check here. Last year, the New York Times published a series of articles which took a more critical look at the advances in personal genomics, and I want to read this out of Andrew Pollack's piece back in the New York Times in June, and then we'll chat about it. A decade ago, drug companies spent billions of dollars equipping themselves to harness the newly revealed secrets of human biology. Investors bid the stocks of tiny genomics companies to stratospheric heights. That genome bubble has long since popped. And not only has there been no pharmacopoeia, but some experts say the Human Genome Project might have, at least temporarily, bogged down the drug industry with information overload. Let me start with you on this. What happened to stall the promise of genomics in new drug development?
I think the fundamental thing that happened, it was enormously overhyped what the Genome Project would do. I mean, really, the Genome Project did two important things. It gave us a list of all of the human genomes, and it democratized them so that any biologist had ready access to any particular gene that they might be interested in. And there was, beyond this, the promise that every gene was a drug. It would be easy to convert genes into drugs. And this was blatant nonsense. I mean, biology is all about, and disease is all about the complexity of biological networks and understanding connections and interactions. It isn't about individual genes. So I think the fault lies partly with some of the people that pushed the Genome Project, but it lies with a lot of investors who just didn't really understand that this was a pioneering effort that transformed biology, that is the Genome Project, but it wasn't a panacea for creating drugs.
Let's talk about that quote some more. Catherine, you want to follow up on that?
Yeah, well, I definitely agree with what was just said. There was a lot of hype and misunderstanding of what one single genome in the Genome Project was going to give us. But make no mistake, the Genome Project and the technology and the computational technology that came out of it has really revolutionized biomedical research. It's given us really incredible tools to go along the pathway to discover those drugs, to make those stratifications of patients that will allow us to treat in a more individualized, more efficient way. The technology has just better and better, and now we're looking at being able to get all the individual sequences of everyone's genomes, do the kind of proteomics you heard about. We're at a level now where we're really well poised, although I say it's research, so it's incremental, poised to make the kind of advances in drugs. That was the promise of the Genome Project. I think we are going to see some of that.
Allison, they talked in the piece there about information overload.
Yes.
Is that an issue? Is that a
Well,
problem?
I think there is. Well, there is an information capacity problem. We don't currently, I don't think, although some of the panel can speak to this, have enough informatics to be able to deal with all the data. If tomorrow we could go out and say, sequence all 13 million Ontarians or whatever we have, and what would we do with it, and where could we store it, and how could we even store it currently in our computers? That we don't necessarily have that. I think the big thing that what happened was that people, the simple stuff was not done, but when we look at some of the drugs where one size fit all pretty well, and so people assumed that when we got the Human Genome Project we'd be able to do that, but the whole promise of the Human Genome Project is to show one size doesn't fit all. Well, that makes it way more complicated. And we also know that everything in that environment, or the whole environment, they also have genomes too. And so we're all interacting all the time. And so that is what's made it much more complicated. But I think people also need to realize that an average drug developed without genomic information, et cetera, that's a 12 to 15 year timeline too. And so we're at year 10. We're probably not 5 years from a whole host of drugs coming along, but we're not. I think it's really that overpromise that happened, as opposed to not being on the right track. I think we are on the right track.
Amy, is too much information a real problem?
Yeah, I mean, I think it's an informatics issue, but I think that can be solved. I think the real issue is an interpretation of the information. We just don't know enough yet, and we have made tremendous progress, particularly on the technology side. It's really kind of boggling to the mind how quickly the technology has advanced. But our ability to understand sort of the data that's being spit out and generated at such tremendous speed and at such low cost has not caught up to that. And so I think that's the biggest bottleneck in sort of translating genomics into a clinical setting, whether it's through pharmacogenomics and drug development, or whether it's through genetic risk assessment and primary care.
Let me follow up on that with a graphic we're going to call All Talk and No Action. And this again is from the New York Times. Sorry, you're going to think we're picking on you here. We're really not. But this is from 6-7 months ago in the New York Times. And Nicholas Wade did it. Here we go, Michael. 10 years after President Bill Clinton announced that the first draft of the human genome was complete, medicine has yet to see any large part of the promised benefits. For biologists, the genome has yielded one insightful surprise after another. But the primary goal of the $3 billion Human Genome Project, to ferret out the genetic roots of common diseases like cancer and Alzheimer's and then generate treatments, remains largely elusive. Indeed, after 10 years of effort, geneticists are almost back to square one in knowing where to look for the roots of common disease. One sign of the genome's limited use for medicine so far was a recent test of genetic predictions for heart disease. A medical team led by Nina P. Painter of Brigham and Women's Hospital in Boston collected 101 genetic variants that had been statistically linked to heart disease in various genome scanning studies. But the variants turned out to have no value in forecasting disease among 19,000 women who had been followed for 12 years. The old-fashioned method of taking a family history was a better guide, Dr. Painter reported this February in the Journal of the American Medical Association. Okay, Lillian, that's a pretty tough assessment, but is that a fair assessment?
I don't think so. I mean, it's fair that in some situations, getting all that information didn't help us move the field very far, but there are instances where we have moved the field. We have now known some very useful drugs to target breast cancer, melanoma, kidney cancer, and that's because we understand more of the cancer genome in these cases. And I think in that case, we are saving lives, or we are prolonging disease control in these cancer patients. I don't think you can apply the philosophy of genomics to all cancer or non-cancer fields and expect we make big improvements in a very quick time. I think that's just far-fetched and not realistic. But I think the whole genome project, Human Genome Project, has raised awareness that we should understand beyond just a microscope. There is more than just what meets the eye. There's the scientific makeup and the coding of the drug or the cancer or the body that's telling us information that we can learn further.
Let me get Lee and then Allison on whether or not that tough
I'd like
quote is
to
a fair
make a
quote.
comment about that if I could.
You're up.
I think the fundamental premise that Wade made is utterly flawed. The idea that the genome promised to solve the problems of medicine and to make drugs is nonsense. I was there in the beginning, and the genome has done an enormous number of things. It gave us a parts list for systems biology. As I said earlier, it democratized genes. It pioneered transformational technologies that have revolutionized all of biology. It brought to biology for the first time computer scientists and mathematicians that were serious in really helping us. It opened up the whole idea that data had to be accurate, the idea that the information should be open to the biological community, and it revolutionized all aspects of biology, of evolution. It's started to pioneer genetic stratification and identification of disease genes and the ability to look at drug responses and things like that. But one of the really critical things I think it did is it opened up the whole realm of big as opposed to small science and demonstrated that there are projects that it is essential to use big science on because they're too complex. They require cross-disciplinary cultures to solve. So I'd say if there's anything that really transformed biology in the last 100 years, it was the Genome Project. And I think Wade and Pollock and the others who take this very narrow, myopic view are really missing a major point.
Allison, if it's in the New York Times, we know it's all the news that's fit to print. But do you think it's accurate? Do you think it's fair?
No, I don't think it's fair. But I think that it brings up one point which I think people lose in genomics is that genomics is, especially if we're talking about medicine, it's one part of the medical system. It's part of the healthcare. So it's still important and going to be incredibly important to take family history. Probably do family genomes like Lee was talking about, but also because that gives you environmental influences on that genetic material that you were going to look at. It has made a huge amount of information available to a lot of people. I think somewhere down the road, very early on, some mistake was made that said we were going to have cures for these diseases. I don't know how that happened. Most of the people I know who worked in the field didn't think that was going to happen that quickly.
Somebody promised it somewhere along the way.
Someone promised it somewhere. Or someone misinterpreted and promised it somewhere.
Or somebody's publicist maybe did.
Something happened. And so I think we need to backtrack and say, look, this is what it's really given us. This is where we really think we are. I think one of the very important things that Lee mentions in his P4 is the participatory part, that it's your genome. It's going to be your part of your medical record, your medical team. And so it's your opportunity to participate in that. And so I think that's very important. And I think that is also something that the human genome has given us and will continue to give us as down the road more and more information about your own personal well-being, etc., as we gather more information. But we just need to continue to work and get more information and be realistic with people. But I think the big science part is really important, because in order to really know what's going on, you need to do a lot of people and a lot of genomes and a lot of information.
Well, let me follow up on that. Last night we had on the guy who's heading up the Ontario Health Study.
Yes.
He's trying to get 1 million people to go online and tell us their family history and all of that kind of stuff.
Right.
Presumably that's what you've got in mind, right?
Absolutely, absolutely. So there's a perfect example in the Ontario Health Study. People can go on. A subset of those people will then do a further informed consent to have further information taken them, including their genetics and genomics. Absolutely, that's the kind of big science that we will need, and that will actually be transformative in Ontario healthcare.
This is a new one. So I've got big pharma and now big science as well.
Big science.
This is a new thing. Okay. The New York Times quote, what do you think?
I don't agree at all. So I would echo with what everybody else said.
Nicholas Wade has no fans here today at all, does he?
No fans whatsoever. I don't really think any of the scientists who did the Human Genome Project promised to cure all disease or the things that were quoted there. They promised to give information that was urgently needed, badly needed, to try and make sense of disease, to try and improve human health. We needed the genome information, and it's been extraordinary. And as I said before, and it's been said by others, the technology has brought this new big science that allows us to ask questions we couldn't ask before about human disease, and to really try and understand pathways that lead to disease and design better medications in a much more logical context than we could before. It's already having an impact on medicine.
Amy, do I need to ask you about the New York Times piece, or you're just going to pile on as well?
No, yeah, I mean, I agree. I think the biggest problem has been sort of the hype around the Human Genome Project, and you know, it's created unreasonable expectations. And so I think it's important for people to just understand the promise of genomics, but also the limitations of the science to kind of be the window into the future, or the sole window into the future.
Okay, let me get a couple of last things on the
Could
agenda
I make
here.
one quick point?
Yeah, surely.
And
We got
that
5
is-
minutes to go here, so
The
go
point I'd
ahead.
like to really emphasize, the point I'd like to emphasize is it isn't just genomics, it's proteomics, it's the analysis of individual cells, it's a whole series of hierarchical levels of information that are going to tell us about disease. And I think this really is the promise for the future of a really deep understanding of the complexity of disease and how to deal with it effectively.
Lee, while you've got the floor, let me ask you to follow up in describing the P4 as you did earlier in the program: predictive, personalized, prevention, and participatory. Aside from the science, what's it going to take to transform the healthcare system so that it really works that way?
I think the key, so I'd say number one, P4 medicine isn't incremental, it's a fundamental paradigm change. And I've been involved in several paradigm changes in my career, and my experience is what convinces people of paradigm changes is that one has to demonstrate the power of the new paradigm. So what I think is going to be necessary, and this is a collaboration that my institution, the Institute for Systems Biology, has with Ohio State Medical School, is we're taking on 2 pilot projects that are going to use these powerful new tools of systems biology, including genomics, but many others as well, to sculpt and determine metrics for wellness. That, I think, is really going to be transformational, and to look at a really difficult medical problem, heart failure. If we succeed in one or both of those in demonstrating the power of this new P4 medicine, then we're going to be in a terrific position, because of a strategic partnership with the state of Luxembourg, to go back there and in a nation, a small nation of 500,000 people with a single payer system, and I've talked to the Minister of Economy and the Minister of Health about this, set up P4 medicine so we can demonstrate its validity on a national basis, and then perhaps the heterogeneous system in the US and certainly less heterogeneous system in Canada are really going to sit up and take care. But I think the key is proving to the world that you can do the many things you've said you can do it. And I have to say, my own estimate is far less than the multiple decades to really achieve first this ability to stratify diseases, that's where it all starts, and then the ability to do the impedance match against drugs and make transformational and revolutionary changes in medicine.
Okay, Catherine, with a couple of minutes left here, are doctors, in your experience, taking to this? Personal genomics, that kind of thing?
There's a lot of interest, but a lot of issues in terms of knowledge transfer and knowledge dissemination. So the whole, all that's been happening in genomic sciences and in informatics has been happening very, very quickly. And I would say in the medical community or in the healthcare community as a whole, there's some really major gaps in knowledge and understanding of the new knowledge, of trying to keep up with it, of trying to understand what it means. And I think, at least in our province, one thing that really has to happen is a huge improvement on both knowledge transfer to the medical community and on collecting the medical data into databases in a much more organized way. Every patient comes into every physician and they collect the information in a kind of a standardized way that you can really...
You sound like you're talking about e-health, which we seem to have had a devil of a time figuring out.
We have had a devil of a time, but we need to get around that problem. We need to have the clinical information in standardized ways that can be used to then look at the genetic or proteomic information in order to put that together and do the very best for our patients. Those are 2 big challenges facing this province.
Lily, let me give you the last word on that.
I completely agree with that. I think making sure that not just the doctors understand the information, but the patients, where the data come from, is understandable to them, I think is crucial. And this is what we need, a good informatic systems as well as understanding the science that we can actually make it happen in the clinic to benefit the care of our patients.
Do you know who TACMAC is?
Yes, I'm married to him. My genes tell me that I should.
Your genes tell you should. He's been on this program, and I don't have to tell you, he's a genius. Anyway, thank you very much, everybody, for this discussion today about personalized medicine. Leroy Hood on the line from Seattle, Washington. Amy McGuire on the line from Houston, Texas. Great to have you two in Points Beyond for our program today. Catherine Siminovich from Mount Sinai Hospital, Lillian Su from Princess Margaret Hospital, Alison Symington, Ontario Genomics Institute. Great discussion, everybody. Thank you very much.
Thank you.